cyclic AMP is activated. We've looked at G proteins and direct activation of calcium channels. No enzyme activation required. Okay? Now we're going to look at the more complicated one, which is G proteins and a membrane-bound enzyme that also involves calcium and protein kinases. So this third one is kind of a combination of the first two as far as the events that occur. All right? This is the diagram you have. I have a couple of modifications to make to it, but we'll take those in stride. So similar to the first component with adenylate cyclase, we have a membrane-bound enzyme that is going to interact with our alpha subunit. And this enzyme is known as phospholipase C instead of adenylate cyclase, but it has the ASE in its name, so you can recognize that it's an enzyme. And it would be inactive on its own in the plasma membrane until our alpha subunit meets up with it. So here you can see it on the screen, phospholipase C, sometimes abbreviated PLC. There's a lot of phosphos and P words in here, okay? So you're going to need to make some effort to keep them straight. Phospholipase C functions to break down lipid molecules that are already present in the membrane. And that's not very clear in this slide. It makes it look like the phospholinositol is coming from the cytoplasm, right? But its substrate is actually one of these phospholipid molecules. So what this works on is one of the lipids in the membrane. And when it comes in contact with the enzyme, then it is broken down. So in lab reports or on the lecture exams, we have a lot of these abbreviations, PIP2, whatever, for full credit, and I have a reminder on the homework that goes through these steps. Write the whole thing out. Okay?
Can you have a legend, like it's, say, the...
Sure. You can put it on the side of the page. You're not writing it over and over and over. Yeah, that's fine. As long as I see on that page somewhere or attached to it that you know what those words stand for, that's perfectly fine. So phospholipase C then chemically, as an enzyme, increases the breakdown, the rate of reaction for phospholinositol. Two molecules are produced. Each of them is considered to be functioning as a second messenger. Right? Even though literature now considers one of those to stay within the membrane. Again, differing from this particular image. So I'm going to keep them in blue. And one molecule of phospholinositol is known as inositol triphosphate, or IP3. This is the calcium side. Remember I said we were combining both aspects of it, protein kinase activation and calcium activation. So this is the calcium side. And inositol triphosphate causes calcium channels in the endoplasmic reticulum, so from inside the cell. The previous step we looked at was calcium from outside the cell comes in. Inositol triphosphate opens calcium channels in similar to a sarcoplasmic reticulum. So stores mostly in smooth muscle. So we're going to release calcium ions into the cytoplasm. Okay, so that's the role of inositol triphosphate. And then these calciums ions can regulate cell activity just like the calciums from outside the cell. So one molecule made from phosphoinositol is inositol triphosphate, okay, or IP3. The other molecule is diacylglycerol. And in this diagram, it looks like it's a free cytoplasmic response. It actually stays within the plasma membrane. And it will activate protein kinase C, which then phosphorylates other enzymes. So again, to reiterate, our first component, we looked at alpha G protein activating cyclic AMP, activated enzyme cyclase, which activated cyclic AMP, and we ended up with protein kinase A. Then we opened calcium channels with our alpha subunit. And now with this pathway, and I know with Bio 430, when we talked about neurotransmitters, I showed you slides of this. I didn't require you to know the IP3 and the DHE, okay, but this is how some of our neurotransmitters work as well. Alpha subunit activates a membrane-bound enzyme, and then we go both pathways. We liberate calcium ions, and we activate protein kinase. So we can phosphorylate other molecules, or we can use calcium ions to activate. But IP3 and DHE, and then also the calcium ions are going to be considered second messengers. But typically in this pathway, these are the molecules that are identified as the second messenger, IP3 and DHE. You seem lighter in here? These lights seem to brighten and dim on their own. So this is a diagram that's not on, I'll get to you in just a second, that's not on, I just got this up here Tuesday, and so you can see, here's the IP3 molecule, made from, the PIP2 molecule isn't up there, well there it is, there's IP3, and it's going to open a calcium channel, all right? And then diacylglycerol is in the membrane, and it's activating protein kinase C. So this is just kind of the same thing that I drew up on the board. Yes, hold on just a second, there was a question.
Why would the calcium also be a second messenger?
Because it's going on to change other behaviors, and it's diffusing through the cell. So just like our original calcium and our second pathway, it doesn't bind to calmodulin always, but it's going to go on to change other activities.
So in the lab, I noticed that it says it asks of a phosphorylation of, and it says blank calcium channels after the DHE.
So I wondered, where are those calcium? Are they membrane? Are they the membranes, or are they in the ER here?
Well, it has two boxes of calcium channels, and you illustrated the one with IP3, which makes sense. But then it says the phosphorylation of extra calcium channels that you didn't demonstrate with the DHE.
Well, that would be the same as the previous slide.
So
with the membrane.
So the DHE also opens calcium channels?
It can through the protein kinase C, but not directly. See me after class on that one.